Hello, my name is Andrew Smith, and I'm a professor and consultant microbiologist at the Scottish Meningococcal Reference Laboratory. My colleague Roisin Ure, highlighted here, is the technical lead for meningococcal sequencing. The aim of this presentation is to give you a very brief overview of how we utilize the MRF MGL in Scotland. The laboratory is based in the new Lister building at Glasgow Royal Infirmary, and this slide highlights some of the details linked to our organisation of the NHS in Scotland. So we have 14 NHS boards with 18 NHS local diagnostic laboratories that send us meningococcal isolates. Now, just to give you an idea of the range of distances that involves, this can vary from 3 meters to the laboratory below us to the Shetland lab, which is about 325 miles away. In terms of other logistics and population served, we have a population of nearly 5.5 million. And since the service has gone live, which we went in April 2018, we've whole genome sequenced some 68 meningococcal isolates. Now, really for historical reasons, the local diagnostic labs send us their meningococcal isolates for initial serogrouping. And this is the process that we undertake for that initial step. So serogrouping and SIAD PCR. The turnaround time for this is in the region of 24 to 48 hours. Here's an example of that initial report, detailing the serogroup confirmed by SIAD PCR. And this can help public health colleagues and consultant colleagues rule in or rule out isolates at an early stage. At the next stage, then, we move on to the sequencing workflow. And once we've got a culture, we then move into the DNA extraction and library preparation stage, and that usually takes 1 day. We then use an Illumina MiSeq. We have one in the department, and this can take between 2 to 4 days. Now, because of the logistics of running the operation in conjunction with other pathogens in the unit, we have a sequencing run that happens on a Tuesday and a Friday. We don't tend to run a weekend service. So moving on to analysis of the sequence and pipelines, that can take another 24 to 72 hours. So in total, we're looking at a time of 4 to 7 days. And of course, we use the PubMLST to interpret those sequences. Here's a hard copy report that goes out to a diagnostic lab that contains pretty basic information. For the hard copy, we just tend to give out very basic typing information. In terms of electronic reporting, this is only available through our local health board through a system called Telepath, which is pretty basic, as you can see. So here we have the meningococcal serogroup. We give out AST data, and that's determined by e-test. And then we give them some typing data, as I've highlighted, the same in the hard copy. But for the electronic copy, there is additional typing information. We also give them access to the whole genome sequence key that you can use to interrogate PubMLST. And the additional information there, you can see I've highlighted, is input to the Mendevar index. So for our public health Scotland colleagues, we provide them with a written communication that highlights Bexsero and Tremember reactivity. We also provide them a nearest match CGMLST allelic difference profile, as I've highlighted on the slide there. So in summary, for our interim report, we provide a serogroup result within 24 to 48 hours. And the final whole genome sequencing report between 4 and 7 days. For our Public Health Scotland colleagues, we will provide details on this in the subsequent presentation. We provide them with the Mendevar index and also the nearest match CGMLST profile. Finally, as ever, it's a team event, and I'd like to acknowledge the input of NHS GG&C colleagues, University of Oxford, and Public Health Scotland.